I will continue about the virtual brain, and I'm particularly interested in the clinical applications. Maybe it fits also because I work as a postdoc in Petra's lab and at the same time also as clinical neurologist. So I really want that this research comes into the clinics and to the patient's bed. And this is just a recap. In summary, what is TVB? I think we can simplify it. We use a network, a structural connectivity, and we use this as a skeleton and bring it to life by mathematics, by a local mathematical model of neural activity in each node. And then we use empirical data of different modalities to fit the data and to validate the model. And then we have built a virtual brain. What we should forget is that this virtual brain is a representation on different scales. It starts from the large scale, like the white matter connections between brain regions, but it goes down to microcircuits, to synapses, and even to subcellular structures. They are just a bit indirectly represented in these mathematical models. And why is this so important? Because when we want to understand disease mechanisms, these typically take place on this molecular level, because typically most neurological diseases have a basis in the area of genetics and proteins. Therefore, I want to take you to a short journey inside the brain on the molecular level and explain how we can make use of this. And therefore, I start with where we come from, with the mathematics. This looks complicated, but actually it's a very general formulation of how we can calculate the activity in a brain network model. So the activity of one node of this model can be given as a differential equation, and the function, it's just a function of the activity of all other nodes, then of the local parameters at this node, of the connectivity that connects the nodes, and of the global scaling factor that scales the connectivity. And why is this important? Because these are the factors where we can inject this molecular information. One of these, for example, by adapting the connectivity by plastic rules, by changing the local parameters, or by insert a perturbation on the activity level of the nodes. And when we come from this, where do we want to go? Into an application, and in my understanding there are three main branches where we can build such a clinical application. We can improve diagnostics by using simulated data for developing new biomarkers, for improving diagnostic procedures. We can test virtual treatments, like, for example, here shown with the virtual deep brain stimulation, a project by Jill Meyer, who will also have a talk tomorrow. But we can also try out virtual treatments on the pharmacological level. And what connects both is the understanding of mechanisms. And why is this so important? Because when we rebuild a disease in a virtual brain, so we have a virtual diseased brain, then we have also all the means of mathematics to understand this object, and therefore we can also understand better the mechanisms underlying the disease. So I will start with an example that merges all these three branches a bit together. It's from my own doctoral thesis on Alzheimer's disease. What we did here is we used amyloid PET data in Alzheimer's disease. So it's amyloid beta is one of the two hallmark proteins in Alzheimer's disease. And what we did is we transferred the local burden of amyloid per region into an impaired excitation-inhibition balance. Basically because it was known that amyloid beta typically affects inhibitory neurons, leading to an excessed excitation, excitotoxic effects, and so on. And what we saw in this model is that phenomena, as we also know from Alzheimer's patients, appeared, basically a slowing from physiological alpha activity to pathological theta and delta activity. And when we then used this artificial data, so it was data on the local field's potential level that was never measured, that was just derived from this model, and we trained a classifier model with it, the performance was better than with the empirical data alone. So we could show that this simulated artificial data can improve diagnostic classification. In the same model, we could also already test out a virtual treatment. Here we used a drug that is already approved for Alzheimer's disease, memantine, which is an NMDA antagonist. It's a symptomatic treatment of Alzheimer's, improving memory function. And that has also shown that it slows down or it reverses the changes in EEG in Alzheimer's patients. And in our model, it was the same thing to see. We saw that the slowing in the Alzheimer group was reserved by reducing the transmission on the NMDergic synapse in the model. So this was where we started actually of this approach to implement molecular information. I was very happy to see later that this model was in different ways reproduced. In 2 times we were involved. Here in the lower panel you can see actually work with the same data set, fitting a different model to the functional MRI data, and the result was that indeed we need this lower inhibition to explain the phenomena we see in the fMRI. We used the same model with different data, not PETs, but simulated amyloid concentrations derived from a protein-protein interaction model, where we also saw this dependence of alpha activity and inhibition. And one work actually where we had nothing to do with it used a completely different model with the same mechanistic function, also showing the slowing phenomenon. So actually this seems to be an interesting, promising avenue in Alzheimer's. Using this as an example, I would go further and say, okay, we can understand the brain or the virtual brain as a mechanistic machine. And if we do this, then we can think about what can we do now in science to make use of it. And I think there are 3 ideas to do so. We can constrain the model by molecular data, to constrain it with more information that is more close to the origin of the disease. We can use the technology to understand the mechanisms better, and we can do classical mathematical modeling and rebuild a mechanism in silico. And I will start to give some examples. Actually, this is ongoing work, so it's not completely finished, but I think already quite interesting to show. So when we constrain this model, of course, we can make use of all this beautiful data that is available. Here's just the eBrains is shown, but I think especially in this workshop we have seen so much data on the very, very micro scale level that contains a lot of information. So of course, when you find a good hypothesis and a good concept for a disease, you can implement this data that is already available. But often we have the problem that the data that we would need is actually not available for a disease because, for example, the technique does not exist. Not every neurological disease has, like with Alzheimer's, an amyloid PET where it can spatially map an underlying protein. But the information is there. We have, for example, knowledge graphs that contain the biological information about the interaction of processes in the disease. Here you see a knowledge graph that describes how different proteins, genes, and chemical compounds in Alzheimer's disease interact and affect each other. But what we do not know is where does this happen and where does this take place? And this is crucial, of course, if we want to implement it into a 3-dimensional brain simulation. To solve this, we were developing a new tool that is still unpublished, but will hopefully soon be out. And there we make use of computational semantics. So we employ a software that was already built before, Skyview, which is actually a corpus of literature in PubMed, but the articles there were annotated with rich metadata information. So the single words are marked with particular meanings, for example, a mesh term or an HGNC identifier for genes and the corresponding proteins, but also anatomical terms. So for each word that refers to neuroanatomy, we have a unique identifier from the Uberon ontology, which is a cross-species anatomical ontology. And because we have this, we can extract statistics about the co-appearance of a search query and an anatomical term, getting to a list which anatomical terms are most frequent and most strongly associated in the whole biomedical literature with, for example, Alzheimer's disease or memory or whatever. What is then missing is to translate this to the brain space. And for this, we have done a quite detailed anatomical curation and manually mapped about 1,000 anatomical terms to a 3-dimensional brain template. We used the parcellation of the Human Connectome project by Glasser et al. And by using this assignment matrix, we can translate the statistics onto a brain map and can use this to infer the models further. I can just quickly show you that surprisingly it works extremely good. So across modalities starting from MEG to receptor densities to cerebral blood flow, glucose metabolism, but also brain functions, this tool can resemble quite well the spatial properties of characteristics of the brain, and therefore is also a very promising tool to go deeper and understand and map mechanisms where we don't have imaging data yet available. So from this, I will go further and show how we can use this to understand mechanisms better. And I start with this ongoing work where we used the TV-based tool that I just showed for the semantic mapping of all genes. We just did it for all genes that we found in HTNC for humans and got a lot of maps. And on the other side, we were collaborating with Munich, and Nikolaos Koutsoulleris provided us with patterns that are distinctive for predicting the onset of psychosis in a population at risk. So he had a volumetric pattern of volume increase and volume decrease that can predict this onset of psychosis, but he had no idea what might be the mechanisms behind. And so we collaborated and we used the support vector regression to reconstruct, like in a puzzle, the pattern. And then we had to look to the genes that were involved. And by clustering them, we can, in that case, figure out 3 main clusters. 2 are not so surprising, so it's neural growth and it's neurotransmitter homeostasis. But 1, for example, is quite surprising, which is the immunological response, especially related to interleukins and tumor necrosis factor. And similar to this, we found a similar finding in Alzheimer's disease. So we did the same mapping with the knowledge graphs that I've shown you earlier with the mechanisms about Alzheimer's disease, and we just extracted the maps that had the most spatial information, just to extract kind of the core spatial vicious circle. And indeed, it was building a circle explaining how driven and actually started by amyloid beta, we can get to a circle between tau protein and inflammatory response and excitation that can maintain a neurodegenerative disease. So we wanted to test if we can also remodel this approach. And for this, we went again back to ADNI data. This is a work together with Luise da Costa-Zemsch, who was actually also here, where we used the same model as I've shown earlier, but in a subcohort of ADNI where immunological markers in CSF were also available. And we wanted to know if there is a coincidence of hyperexcitation and hyperinflammation. And we looked into tumor necrosis factor levels. And indeed, this was the case for the group of mild cognitive impairments. And actually, it was only for the mild cognitive impairment group the case, not for Alzheimer's, not for cognitive control, cognitive normal controls. And this goes along with the hypothesis that in the early phase of Alzheimer's disease, there is an excitotoxic inflammatory phase that then afterwards goes into the neurodegenerative decline. And when we then analyzed the mechanisms further in the gene ontology Go, we were also able to see that most connections from immunological markers to excitation go through this TNF molecule. So actually this hypothesis makes a lot of sense. And when we compared the same model with all the available immunomediators here, then we also saw that indeed TNF-alpha has the second highest excitoinflammation here. So this is also a way for a complete new hypothesis generation with a just very unusual approach. So finally, I will also go to the area of how to rebuild these mechanisms properly, because when we do all this, it's of course of crucial importance that this rebuilding modeling task is done properly. And I provide you with some examples. I showed you actually an intermediate complicated model so far. This is the Jans-Ritt model with 3 populations and quite a lot of parameters already. There are very detailed models of neural activity available. For example, this is the Lata-Breakspear model, and it has extremely detailed local parameters describing ion channels and their properties, so you can in a very detailed way model the effects of calcium, potassium, and sodium. What was not available yet was an implementation in the mouse. So we did this because especially this molecular data on the ion level is usually more available in the mouse, and we were happy to see that we could reproduce functional connectivity in the mouse, but also show that each of these ions can moderate the behavior, and especially in this model, it's very interesting because it's a chaotic oscillator model, so it changes its behavior significantly from an oscillatory to a chaotic regime. And we were also able to show that especially calcium can heavily modulate the way how the model reacts to stimuli from outside. We also went further into the modulation of NMDA receptors. I showed you earlier a very simple way of modeling NMDA receptor transmission. Another PhD student, Jennifer Tham, has done this very intensively because she took into account that, of course, we have NMDA receptors not only on pyramidal cells, we have NMDA receptors also on inhibitory cells, and actually the effects of NMDAergic transmission on excitatory and inhibitory cells are interacting and partly also counteracting themselves. And therefore, we have to take into account all of them. And when you do this, we can indeed see phenomena that are well known from a quite pure NMDAergic antagonism, which we see in ketamine narcosis, which is an increase of firing rate, while at the same time the frequency shows a slowing, basically again, decrease of alpha and an increase of theta. And this we were able to reproduce with this model. We also wanted to see if we can close the loop to the molecular scale. What do I mean with this? Typically, the outcome of the virtual brain is either something electrophysiological, so EEG, MEG, or BOLD as a fMRI equivalent. But we wanted to know if we can also calculate the glucose metabolism, like in FDG PET. And for this, we employed a neurogliovascular model of how ATP is exchanged between neurons, glia, and blood vessels, and implemented it into TVB and fitted it with static and functional. Actually, we validated it, we fitted it with static PET and validated it also with functional PET. And this is extremely interesting because by this we can see the differences how glucose PET reacts in a different way as a response function than fMRI. And why is this so interesting? Because both are measuring the same thing. Both are indirect measures of the actual brain activity. And when we use two methods that have a completely different way to measure something, we can hopefully understand better the underlying activity itself. Here we were able to get quite good fits for the static PET and also for the functional PET, although this is a method that is not so commonly in use yet. And we were even able to quite precisely explain why and also how much the metabolism of the brain is slower or how much the brain needs less energy in sleep or in coma compared to the state of wakefulness just by applying this biologically based model to two different oscillatory regimes. Finally, I just want to sum up that while we do all these things with the virtual brain, it's of course extremely important to do it in a transparent way, because I've shown you now some ideas how we can implement disease processes, and of course the ultimate validation is if it works then in the end as a clinical application, but such decisions should be reliable and justifiable and at the same time also reproducible. And for this we were in another project with a PhD student, Leon Martin, developing an ontology of the virtual brain, describing all the mathematical elements in it and also their relationship with each other. And this ontology is also linked to biological concepts from the gene ontology and therefore offers a completely new and reliable infrastructure to also store and collect the metadata of simulations and makes them hopefully much more reproducible. So to sum up, I presented you some ideas how to bridge the scales from molecules through the brain, hopefully up to the level of behavior and disease. And personally, I believe that when we are open-minded and use all the tools that we have in computational research, we can really come up with applications that come into clinics. And with this, I thank you for your attention and my collaborators from the lab, and yeah, finish my talk.
Thank you.